DNA, or deoxyribonucleic acid, is the fundamental blueprint of life, a complex molecule that carries the genetic instructions necessary for the growth, development, functioning, and reproduction of all known living organisms. At its core, DNA is composed of units called nucleotides. Each nucleotide consists of three components: a phosphate group, a sugar molecule, and a nitrogenous base. The sugar in DNA is deoxyribose, and the nitrogenous bases come in four varieties: adenine, A, thymine, T, cytosine, C, and guanine, G. The sequence of these nitrogenous bases encodes the genetic information. Nucleotides link together through covalent bonds between the phosphate of one nucleotide and the sugar of another, forming a long chain-like structure known as a polynucleotide. This chain then twists into a double helix, a shape famously characterized by James Watson and Francis Crick. The two strands of the double helix are held together by hydrogen bonds between the nitrogenous bases, where adenine pairs with thymine and cytosine pairs with guanine. This pairing is crucial because it ensures that the genetic information can be accurately replicated and transmitted during cell division. When a cell needs to produce a specific protein, the process begins with transcription, where a segment of DNA unwinds, and an enzyme called RNA polymerase synthesizes a complementary strand of messenger RNA, or mRNA. This mRNA strand is essentially a copy of the gene that encodes the protein. Unlike DNA, which remains in the nucleus, mRNA is designed to leave the nucleus and enter the cytoplasm, where it will be translated into a protein. As mRNA exits the nucleus, it undergoes several modifications, including the addition of a 5-foot cap and a poly-A tail, which help protect it from degradation and assist in its recognition by the ribosomes. The ribosomes are the cellular machinery responsible for protein synthesis, and they read the mRNA sequence in sets of 3 bases known as codons. Each codon corresponds to a specific amino acid, the building blocks of proteins. Transfer RNA, or tRNA, plays a vital role in this process. Each tRNA molecule carries a specific amino acid and has an anticodon region that pairs with the corresponding codon on the mRNA strand. As the ribosome moves along the mRNA, tRNA molecules bring their attached amino acids to the growing polypeptide chain, linking them together through peptide bonds. This continues until the ribosome encounters a stop codon, signaling the end of protein synthesis. Proteins, once synthesized, fold into unique 3-dimensional shapes that determine their function within the cell. The diversity of protein functions is staggering. They serve as enzymes that catalyze biochemical reactions, structural components that provide support and shape to cells and tissues, transport molecules that carry substances across cell membranes, and antibodies that defend against pathogens, among many other roles. The specific sequence of amino acids in a protein, dictated by the mRNA, ultimately determines its structure and function. For instance, enzymes like amylase break down starches into sugars, while hemoglobin in red blood cells binds oxygen and transports it throughout the body. The intricate interplay between DNA, mRNA, and proteins illustrates the central dogma of molecular biology. DNA is transcribed into RNA, which is then translated into protein. In summary, the structure of DNA, with its nucleotides and double helix formation, serves as the foundation for the flow of genetic information. mRNA acts as the messenger that carries this information from the nucleus to the ribosomes, where proteins are synthesized. The vast array of functions that proteins perform is essential for the maintenance of life, highlighting the elegance and complexity of biological systems. Understanding these processes not only reveals the intricacies of life at a molecular level, but also underscores the interconnectedness of all living organisms through the shared language of DNA.